Hello all. In this video, we are going to study about the basis of DNA fingerprinting. Basis of DNA fingerprinting. Basis of DNA fingerprinting. So DNA fingerprinting is a technique to identify the similarities and differences of the DNA fragments of two individuals. So it's a technique to find out the similarities and dissimilarities of or between the DNA fragments of two different persons, DNA fragments of two different persons. So DNA fingerprinting, and this DNA fingerprinting technique was for the first time developed by Alec Jeffreys. In the year of 1985. So now we are going to study about the basis of DNA fingerprinting. In general, the DNA carries some non-coding sequences also along with coding sequences, right? In eukaryotes, the DNA carries coding and non-coding sequences. Okay. So generally we have studied about the coding sequences, which consisting of coding genes called exons, and the non-coding sequences we studied about introns like that. So the human DNA is carrying both the coding sequences and as well as non-coding sequences. And these non-coding sequences in the human DNA, the non-coding sequences are nothing but repetitive sequences. The non-coding sequences in the human being are nothing but repetitive sequences, and they are also known as variable number of tandem repeats. These repetitive sequences are also called variable number of tandem repeats. V for variable, N for number, T for tandem, and R for repeats. Variable number of tandem repeats. And this size of VNTR is size and presence of VNTR is specific from person to person. This VNTR is different from person to person. It is very unique. Okay, so because of this, due to this VNTR only, this forms the basis of DNA fingerprinting. This VNTR forms the basis of DNA fingerprinting, and the size of VNTR also ranges between 0.1 kb to 20 kb. The size of VNTR ranges from 0.1 kb to 20 kb. Okay, and this repetitive DNA can be separated from the bulk genomic DNA. This repetitive sequences from the bulk genomic DNA. From the bulk genomic DNA, we can separate this repetitive sequences by using a sequencer. But before that, we have to separate based on their gradient. Okay, density gradient centrifugation we have to perform. Based on that, we will separate them. Okay. So the process I will explain now. How we can separate these repetitive sequences from the bulk genomic DNA. I'll tell you the process now. So based on this, repetitive sequences can be separated from bulk genomic DNA based on density gradient. based on density gradient we can separate. And by using the centrifugal apparatus, the high sedimented material will be the high density material will be sedimented and the low density materials will be supernatant. So in this, by using centrifuge we can separate this particular the repetitive sequences, and the bulk DNA forms the major peaks. But after centrifugation, the bulk DNA will forms the major peaks. Okay, and this repetitive sequences will form minor peaks. Repetitive sequences will form minor peaks. The bulk DNA will form some major peaks, and the smaller and repetitive units will form some minor peaks, and this minor peaks which consisting of a DNA that is called satellite DNA. This particular DNA is called satellite DNA. Okay, so hope you understood. The repetitive sequences can be separated from the bulk xenomic DNA by which gradient? Density gradient. And the bulk DNA will form some major peaks, and the repetitive will form some minor peaks, and those repetitive sequences which forms minor peaks are nothing but satellite DNA. Okay, and this satellite DNA is further classified into many categories like microsatellites and mini satellites based on the base composition. The satellite DNA further classified into microsatellites and mini satellites. Okay, based on the adenine thymine rich or guanine cytosine rich composition. Okay, based on adenine thymine rich or guanosine cytosine rich composition. So based on that composition, base composition, we can again divide them into categories like microsatellites and mini satellites. Okay, and also length of the segment and number of repetitive units also we can take other factors. Okay, number of segments and also number of repetitive units. Based on number of segments and number of repetitive units also we can separate them. Okay, the main thing is based on adenine thymine rich or guanine cytosine rich, who are isolated microsatellites and mini satellites can be isolated or separated. Okay, any difference, you take any difference in the nucleotide sequences like inheritable, observed in the population is called DNA polymorphism. Any difference, whatever the difference that we observe in the nucleotide sequence in the population, that is called DNA polymorphism. And this polymorphism is higher. The polymorphism is higher in the case of this non-coding sequences, repetitive sequences. The DNA polymorphism is the most important characteristic feature of this repetitive sequences, okay, or non-coding DNA sequences. Okay, because the mutations in these sequences may not have any immediate effect. Even the mutations may occur, but they may not show any immediate effect in the individual. Okay, or in case of reproductive ability also. So these mutations will accumulate in the generations after generations and causes polymorphism. The polymorphism is not all of a sudden. Even though the mutations takes place, they may not have immediate effect. So that's why it leads to accumulation of the characters, and as a result, it leads to polymorphism. And this DNA polymorphism of the repetitive sequences will forms the basis of the DNA fingerprinting. Okay, and for even this DNA polymorphism is also essential for the evolution and speciation. Right, this DNA polymorphism will plays the most important role. Okay, so thank you very much.